Hello students, welcome to today's lecture. Before starting today's lecture, we will recall our previous lecture points. In previous lecture, we viewed that there are similarities and differences in male and female reproductive system in terms of organs and functions. However, the reproductive system of male and female work together to create a life. Here, I have stated certain objectives that you should achieve at the end of this lecture. The first is you will able to describe the structure of the male reproductive system. The second is you will able to describe the relative structure of male reproductive system, and you will able to apply the knowledge of anatomic structure to analyze clinical situation and therapeutic application. Today we will go in depth on the topic of male reproductive system. As we explore this topic, we will uncover the remarkable structures and functions that contribute to the reproductive capabilities of males. Let us begin our exploration of the role of male anatomy in reproduction. For better understanding, I have divided the male reproductive organs into two parts: internal organ and external organs. Internal male reproductive organs are not visible, whereas external we can visualize. View the internal and external male reproductive organ list. Do you remember we learned about all these organs in brief in our introductory lecture? However, we can again classify these male reproductive organs into two, that is primary and secondary reproductive organs. According to you, which are the primary male reproductive organs? Testes are primary reproductive organs. Other than testes, all organs are called as secondary reproductive organs. Being a primary male reproductive organ, first we will focus on testes and then we will move to other male reproductive organs. In utero, the testes develop within the abdominal cavity of the fetus and then move down through the inguinal canal into the scrotal sac. The testes are suspended in the scrotal sac where they hang one on either side of the penis, and one often hang lower than the other, and this is a very normal finding. The temperature within the scrotum is about 96 degree Fahrenheit, which is slightly lower than the body temperature, and this is necessary for the production of viable sperm. Let us see the anatomy of testes. Each testis measures approximately in size, that is 4 to 5 centimeter in length, 2.5 centimeter in width and 3 centimeter in thickness. Testes are oval in shape and little bit laterally compressed. Talking about location, already we have discussed that the testes lie in the scrotum outside the pelvic cavity because high intra-abdominal temperature is not suitable for production of normal motile sperms. Now we will talk about the oats of the testes. The testes has three coats, that is tunica vaginalis, which is the outer, inside tunica albuginea, and the third one is tunica vasculosa. The tunica vaginalis is derived from peritoneum, Tunica vaginalis covers the testes on all sides except the posterior border. Tunica vaginalis has two layers, visceral layer and parietal layer. Between these two layers, there is a small amount of serous fluid that prevents friction and allows the testes to move in the scrotum. Tunica vaginalis covers tunica albuginea except at the point of attachment of the epididymis to the testes and along its posterior border where the spermatic vessels enter the gland. Tunica albuginea is a thick, fibrous coat composed of irregular connective tissue that covers the testes internally to tunica vaginalis. This coat sends delicate connective tissue septulae in the interior of testes to divide it into 200 to 300 compartments, that is lobules. The tunica albuginea is thickened along the posterior border of testes to form the mediastinum testis that contains the branches of testicular artery, venous plexus that will continue and pampiliform plexus, rete testis that is a plexus of efferent tubules that conducts the sperms, lymphatic and sympathetic plexus. Tunica vasculosa is the third layer. It is formed by delicate connective tissue. It is the vascular layer of the testes consisting of a plexus of blood vessels that are held together by delicate areolar tissue. It lines the interior of the lobules, that is compartments of testes. In the previous slide, we learned about the outer side of the testes. Now we will see the inner structure of testes. This is the sagittal section of the testes. Tunica albuginea is the covering on the testes which extends into the testes, forming septa that divide the testes into compartments. The testes has about 200 to 300 compartments called as lobules. Inside each compartment is a collection of tightly coiled hollow tubes called the seminiferous tubules. Each lobule contains 1 to 3 seminiferous tubules just visible to the naked eye as delicate threads. Each tubule's length is 70 to 80 centimeter and diameter 0.01 to 0.13 mm. These tubules have a combined length of approximately 250 meters. Each seminiferous tubule is U-shaped and connected to single short straight tubule that enters the mediastinum of the testes. Straight tubules are extensively interconnected, forming a maze of passageways known as the rete testis. After sperm is created in the seminiferous tubules, sperm cells travel towards the epididymis through the rete testis. The rete testis help to mix the sperm cells around in the fluid secreted by Sertoli cells. Before sperm can get to the epididymis, they cannot move. Millions of teeny projections in the rated testes, known as microvilli, help them to move along to the efferent tubules. 15 to 20 large efferent ducts are a series of tubes that connects the rated testes to the epididymis. These ducts are lined with hair-like projections called cilia. Along with a layer of smooth muscles, cilia help to move the sperm into the epididymis. There are three types of cells located in the seminiferous tubules. The first one is Leydig cells. Second one, germ cells and Sertoli cells. There are spaces located between the tubules. In these spaces, there are cluster of cells called the Leydig cells. They are also called as interstitial cells. These cells synthesize and secrete the hormone testosterone at the time of puberty. The testosterone regulates the spermatogenesis and development of secondary sex organs. The testosterone also influences brain and it is responsible for the aggressive behavior and positive attitude of the males. Other two cells are germ cells and Sertoli cells. They are in the seminiferous tubule. Germ cells, also called as spermatogenic cells. The germ cells produce spermatozoa by progressive differentiation. These cells, which are at near the basement membrane of seminiferous tubule, are called as spermatogonia. These cells are at various levels of maturity. The third cells are Sertoli cells. They are also called as sustentoocytes. These cells surrounds the spermatogenic cells and they provide mechanical support and nourishment to the spermatozoa. Sperm cells in their various stages of the development are stored in the spaces between the Sertoli cells. We have to talk about one more type of cells, that is myoid cells. Human seminiferous tubules are surrounded by several layers of smooth muscles like myoid cells. Contracting rhythmically, these may help to squeeze the sperm and testicular fluid through the tubules and out of the testes. Now, let us see the blood supply of the testes. The testicular artery is a long, slender branch from abdominal aorta arising a little below the renal artery. It is the main artery supplying the testes. The artery to the vas deferens is a branch of inferior vesical artery, has an anastomosis with the testicular artery. The testicular vein forms a network around the testicular artery called a pampiliform plexus. This network of vein absorbs heat from the artery before the blood enters the testes, and they maintain the thermoregulation to maintain the environment at a 3 degree less than body temperature that is required for spermatogenesis. At the deep inguinal ring, the venous plexus give rise to a single testicular vein that ascends by side of a testicular artery. Right testicular vein joins inferior vena cava, left testicular vein ends in the left renal vein. The lymphatic supply of the testes involves multiple lymph nodes and vessels. The lymphatic drainage of the testes primarily occurs through two pathways, the superficial lymphatic vessels and the deep lymphatic vessel. The superficial lymphatic vessels arise from the tunia albuginea, and the deep lymphatic vessels arise from the testicular parenchyma. Both the superficial and deep lymphatic vessels eventually reach the para-aortic lymph nodes, also known as the lumbar lymph nodes. From the para-aortic and iliac nodes, the lymphatic drainage continues to the thoracic duct or right lymphatic duct, which eventually returns the lymph to the bloodstream. The testes receive its nerve supply from the autonomic nervous system. The nerves that innervate the testes include sympathetic and parasympathetic fibers. The sympathetic nerves that innervate the testes originate from the thoracic sympathetic chain ganglia, specifically from the T10 to L2 level of the spinal cord. The sympathetic nerve primarily regulate the smooth muscle functions within the testes, such as the contraction of the ductus deferens during ejaculation. Parasympathetic innervation of the testes is derived from the pelvic splantelic nerves, specifically from S2 to S4 level of the spinal cord. These parasympathetic fibers travel through the pelvic plexus and reach the testes. Parasympathetic stimulation is involved in the regulation of blood flow and erection within the testes. It is also important to note that while the autonomic nervous system provides the primary innervation to the testes, there are also sensory nerves present that transmit pain and other sensory information. These sensory fibers are carried by the ilioinguinal and genitofemoral nerves, which are branches of the lumbar plexus. These nerves can transmit pain sensation in conditions such as testicular trauma or inflammation. So in brief, we can say the testes receive both sympathetic and parasympathetic innervation, which play the role in the regulation of smooth muscle function, blood flow, and sensory perception within the testicular region. During embryonic life, the testes develop within the abdominal cavity but descends into the scrotum prior to birth. If they fail to do this and the condition is not corrected, may cause infertility is likely to follow and the risk of testicular cancer is increased. This condition is called as undescended testes or cryptorchidism. Hydrocele. It is the accumulation of the fluid within the tunia vaginalis. The fluid may collect following inflammation of the testes because tunia vaginalis is closely related to the front and the side of the testes, but most hydroceles are idiopathic. The fluid from tunia vaginalis can be removed by inserting a fine trocar and cannula through the scrotal skin. Tumors of the testes. There are two main varieties of testicular tumors are seminoma and teratoma. These tumors are very rare, but there is high risk seen 20 times greater in people with a history of late descent or undescended testes. Most cancer arise from spermatozoa producing cells. The cancer cells from testes spread upward via the lymph vessel to the lumbar lymph nodes at the level of L1 L2 vertebra and produce secondary tumor in the abdomen. Varicocele is a varicose vein in the pampiniform plexus that disturbs venous return from the testicle and may cause a sperm abnormality. Testicular torsion or rotation of testes may occur usually externally. The condition leads to compression of testicular vessels and may lead to ischemic necrosis of testes. The torsion may be relieved by medial rotation of testes. It will be done by external pressure on the scrotum very gently. Here we have finished with the anatomy of testes and its clinical application. Now it's the time for self-assessment. So let us start.